Okay guys, so I'm going to go through the Chapter 7 test review and explain all of the questions and maybe give you a little bit of some extra help to prepare you for the test. Okay, so watch this whole video. All right, so number one, looking at a cell under a microscope, you note that it is a prokaryote. How do you know? Well, guys, the main difference between a prokaryote and a eukaryote is the fact that a prokaryote does not have any organelles. Now, that does include a nucleus, so they do not have a nucleus. They don't have any of the other organelles as well that we talked about. So they don't have an endoplasmic reticulum, they don't have vesicles, they don't have anything. Their DNA is free-floating, and their ribosomes are free-floating. So the answer is C. Number two, which of the following is not found in the nucleus? Okay, so in the nucleus, guys, you've got a nucleolus, that's the dark center. You've got chromatin. Remember, chromatin are your unorganized DNA. They're just the little strands of DNA that you find in your nucleus. And then DNA, which chromatin are. So your answer would be A. Number three, which organelle breaks down organelles that are no longer useful? Lysosome, that's just definition. Your Golgi apparatus, right, transports proteins from, the proteins came from the ER. They went through vesicles to your Golgi apparatus, then your Golgi apparatus modifies them and then spits them out, and they can either go through, in vesicles, they can either go to your cell membrane or they can go to other parts of your cell. Your endoplasmic reticulum, if it's rough, it has ribosomes on it and therefore makes proteins, or it's smooth and produces lipids. And mitochondria, finally, takes sunlight and converts it to food for the plant. So your answer is B. 4. Which sequence correctly traces the path of a protein? Well, I just said it. You go from what makes the protein, which is a ribosome. Then the endoplasmic reticulum, guys, creates that little vesicle that takes the protein from the endoplasmic reticulum to the Golgi apparatus. The Golgi apparatus then will modify your protein. Number 5. Cells that have DNA as their genetic material include, well, both prokaryotes and eukaryotes have DNA. It's just that eukaryotes have a nucleus that contains the DNA, while DNA in prokaryotes is free-floating. All right, so 6. Looking at this, number 1, these are your lipid tails. So these are lipids. They are hydrophobic, meaning water-fearing, and so they're going to turn towards each other because what's out here is water. So they're going to go towards each other because they are averse to water. They don't like water. Number 2 are the phosphate heads. They're your heads right there. And they are hydrophilic, meaning they're water-loving, so that's why they go right up against the water. So outside of the cell and inside of the cell has a lot of water. And then finally, this thing in the center, this is a membrane protein. Okay, so that membrane protein can either be a pump in active transport, or it can be a channel in passive transport. So just keep all of those concepts straight in your head. So also remember the fact that this is your phospholipid bilayer. It not only includes this phospholipid bilayer, these lipids, but it also includes proteins, like this protein, either channel or pump, depending on whether it's passive transport or active transport. All right, 7. Which of the following is a function of the cell membrane? Okay, it does not break down lipids. That would be a lysosome. It does not store water, salts, and proteins. That is a vacuole. It does not keep the cell wall in place, really. I mean, the cell wall doesn't need to be kept in place. It is right behind the cell wall, but it does regulate the movement of materials into and out of the cell. Okay, and the process of that regulation is called homeostasis, the fact that you have a very distinctive inside composition of the cell versus outside. That's homeostasis. All right, the cell membrane contains channels and pumps that help move materials. What are they made up of? Proteins. Remember up here, these are proteins, these are your lipids, so the cell membrane is made up of lipids and proteins. All right, 9. Which means of particle transport requires input of energy? That's active transport. Facilitated diffusion, diffusion, and osmosis, those are all passive transport and don't require energy. All right, the cells of unicellular organisms, we talked about this last class. They cannot be specialized. Cells of multicellular organisms can, like in us. Our muscle cells are very specialized, so they have extra mitochondria in order to produce extra energy, extra ATP, because your muscle cells need a lot of energy. But unicellular cells, they can't be specialized because they're having to deal with the whole world around them. They're not in one specific organism that can specialize based on their function. So C, they have to be able to carry out all the functions, while in a multicellular organism, each cell can do different roles and therefore work kind of like a machine. All right, so here's your diagram, guys. And this was a little bit messed up, so that's why I wanted to go through it. Number 1, there's just a little circle, so it's not clear whether it's a vacuole or a lysosome. On the test, I'd make it more clear, but I just put vacuole and lysosome. Okay, so a vacuole, F, stores food, water, and waste. And a lysosome, G, breaks down larger food molecules. Okay, number 2 is going to be your cytoskeleton. Now notice 2 and 8 are cytoskeleton. What I was wanting, and I'm not going to require this on the test, but what I would like is for you to recognize, since these are so thin and they're less like chunky, this would be microfilaments because they're made up of actin. These would be microtubules because they're made up of tubulin. So if you're not remembering this stuff, make sure to go back and review. But yeah, this is tubulin, microtubules made up of tubulin, microfilaments made up of actin. Okay, but I just wrote cytoskeleton, that's fine. And the function is J. They're responsible for the shape, right, the strength, and movement in cell. Remember, they not only help with the little vesicles moving from the Golgi apparatus to the cell, remember the walking along, but they also can form flagella, those long tails, or the little tiny hairs, which are the cilia. Okay, and that is in prokaryotes that they do that, make the flagella and have cilia. All right, number 3, that is the thing that surrounds your entire nucleus is your nuclear envelope, and it, O, is a phospholipid bilayer, and it surrounds the nucleus. Okay, the nucleolus is in the center, okay, and it makes the ribosomes. Okay. Your nucleus is the, hold on one sec, sorry. Your 6 is the nucleus. The whole thing is called the nucleus, okay? And it contains the hereditary material of the cell. That's DNA, and we'll be studying that in a couple of chapters. 8, again, is the cytoskeleton. I had said microtubules to be exact, but cytoskeleton is fine. 9 is mitochondria. Guys, mitochondria take energy and make it into usable energy, specifically ATP. So if you remember the video of active transport, where the ATP stuck to the protein pump, and then the protein pump finally opened up to allow the sodium and the potassium to move through. That was because the ATP. That is produced in mitochondria, and we're going to be studying that in chapter 9. The smooth ER is here. Notice it has no ribosomes on it. This is the smooth ER, and it creates lipids. Okay, so K makes lipids. Cell membrane is 11, and it controls what goes in and out of the cell because it's made up of lipids, and then it has protein channels and protein pumps. 12 are ribosomes. They can be free-floating or they can be bound to the endoplasmic reticulum, okay, and they make proteins. Golgi apparatus is right here. Remember, your protein goes from the ribosomes that make them, then the ER splits them and puts them into a little vesicle, and the vesicle travels up to the Golgi apparatus. The Golgi apparatus modifies them and then puts them into another little vesicle, and then they can be transported out of the cell. Okay. So I is your answer for that one. Modifies, then packages and transports proteins. Cytoplasm, D, is a clear, thick, jelly-like material that supports organelles. That's all of this. It's mostly water. Vesicles store and transport materials. Okay, so vesicles, that's here. It's showing it budding off of the Golgi apparatus right there. And then the rough ER, which has the ribosomes on it right there. Okay, alrighty. We're going to go move all the way down. Is the cell in the figure to the right a prokaryote or eukaryote? Explain how you could tell. So if you look at this, what you notice is that the DNA is free-floating, the ribosomes are free-floating, and you've got no organelles. So automatically in your head, you should think it's a prokaryote. And the way you explain it, you say, number 1, it has no nucleus, it has no organelles, and the DNA is free-floating. So that's all I wrote. You can include even more. Notice also I wrote that it has a flagella. That's a defining feature of some prokaryotes. Okay, but the main thing, guys, is that its chromosomes are free-floating. It's not in a nucleus, and its ribosomes, all of them are free-floating. They're not. Some of them aren't stuck on an ER because they don't have organelles. How are endocytosis and exocytosis similar? How are they different? So they're both responsible for the mass transport of materials. That's bulk transport. The reason why they are different, however, is because endocytosis, what happens is that the material is going to be entering, right? So the cell membrane goes all the way around the materials that are outside the cell and brings them in as a vesicle. Okay. In contrast, with exocytosis, the membrane of the vesicle fuses with the cell membrane and therefore lets them out of the cell. And so if you guys forgot about how exactly that looks, you can just Google it, and you'll be able to see really, really quickly what it looks like. And so that'll help a lot. All right. For the next one, which type of microscope can produce 3D images? Remember, so I'm going to go over a couple of differences really quickly. Transmission electron microscope, so this is creating a 2D image. This is an electron microscope, so your sample is dead. Scanning electron microscope, you're just scanning the outside of the sample, okay, with electrons, so therefore it'd be 3D, so that's your answer. The answer is B. Okay, let's really quickly talk, we didn't really talk about a simple light microscope. The compound light microscope, guys, is what you guys worked with. Let's really quickly talk about who discovered cells and with what. So remember, the microscope was vital for this discovery of cells. And remember that the person who discovered them was a guy named Hooke. Okay, so Hooke discovered cells. He was looking at cork cells. Remember the whole story of he was looking at the cells and he thought that it looked like the little chambers where monks stayed in a monastery, and they called them those cells, so therefore he called the cork little chambers cells as well. All right. So in order to be able to see all the cells and all of those characteristics that we take for granted now, Hooke's work and the help of the microscope was vital for that. All right, which of the following organisms are prokaryotes? Plants, animals, and fungi are all eukaryotes, guys. So bacteria are prokaryotes. During diffusion, when the concentration of molecules on both sides are the same, the molecules will, well, if they're equal on both sides, guys, what you have is a situation that's called equilibrium. Okay, so if they're equal on both sides, that doesn't mean nothing's going to move. They're going to be moving, but there isn't going to be a net movement, meaning nothing's going to be moving completely to the right or everything's going to be moving to the left. Things are moving equally to the right and to the left at the same time. And the answer, therefore, is C. Which term describes the relatively constant internal physical conditions of an organism? Meaning in a cell, we keep a certain condition despite the fact that outside the cell, it's totally different. And that's homeostasis. Okay. This was a really great question, guys, and I'm going to have you continue to read stuff like this. Tay-Sachs disease, what happens is that this lipid, gangliosides, they build up in the brain and cause death in really young children. And so what I wanted you guys to find out was how this relates to what we've been learning. So if you didn't do this, close this really quickly, pause it, try it on your own, because it's really useful for you to be able to relate what we learned to what you read in papers. If you did do it, listen and find out more. So the organelle that breaks down the gangliosides in normal cells, right? So if you have extra lipids, what would break down those gangliosides would be the lysosomes, because the lysosomes, right, are the cleanup crew. They're the garbage men. So they break down old extra molecules or organelles or whatever you want to get rid of. But in this disease, they don't work. Now, the reason why is because the enzyme that is in the lysosome, the ganglioside, doesn't work. Now, the organelle where the gangliosides are made in the first place, since they're a lipid, would be the smooth ER. So here are two questions you guys can totally answer with your prior knowledge. Now, C refers back to a last chapter we worked on. Explain why ganglioside is unable to break down gangliosides. Now, this is an enzyme. So I drew an enzyme, okay, and this is the substrate, ganglioside. So it needs to fit in perfectly in order to be dissolved in the lysosome. This is in the lysosome. This goes into the lysosome and is dissolved by this enzyme. Now, what happens in Tay-Sachs is that the shape of this ganglioside changes. So maybe it looks like this. Now notice the ganglioside no longer can fit in there, right, at this active site, it no longer can fit. Therefore, the gangliosides in the lysosome can't break down the lipid, and so you have accumulation of a lipid, which then kills the kid eventually. Okay, so review your notes, guys. That's all